Okay, so the last speaker before the coffee break is Anastasia Zhuravleva from University of Leeds, and she's going to talk
about unfolded protein response. So thank you very much for inviting me. And today, I mean, I'm good that I'm the last one because everyone already explained before me, so I don't need to do a lot of theory. So I will discuss some of similar topics as well. So what I'm going to do today, I'm going to talk quite a lot about challenging system, and I'm going to do about what we can do with signaling pathways, right, and how we can understand them better and how we can move from individual snapshot to molecular mechanism, actually, right? And what my lab is interested in is stress. And the good news for everyone, the stress is not only just human problem. And if you think about a protein, which is here, right? So you probably all know this nice schematic presentation of cell. So what is inside the cell? But if you think about the small protein inside the cell, so you can see it's a very, very difficult environment, right? To be folded and to keep and to be folded, right? So because there are a lot of other proteins around, right? So it's a lot of macromolecules like DNA, lipids, small molecules. This environment changes constantly, right? So cells kind of now it's a very difficult task, and this is what we're interested in, how it's actually even possible that proteins keep folded inside the cell, right? And we're particularly interested in molecular chaperones, and molecular chaperones are molecular machines which help other proteins to fold. But we're also interested in unfolded protein response, and I will discuss this in a bit more details later. And of course, we're interested in method development as well, because all this system are very challenging from biological viewpoint, but it's also from technical viewpoint. So we're interested in method development as well. So just to give you a very brief introduction into unfolded protein response, because it's a very complex, I don't have time to explain it in details, but what I want to say, it's a stress response. It's one of the key stress responses inside the cell. It's a stress response inside the endoplasmic reticulum. And the plasmic reticulum is a very unique organelle which is responsible for folding and maturation of 30% of all proteins, right? And these proteins usually membrane proteins and secreted proteins. So in a half an hour, you will go outside, you will eat some cake, right? So your glucose level will increase. You will need to produce insulin, right? So your cells will be in, your beta cells will be in a lot of stress, right? So you will need to produce a lot of chaperones, right? And your unfolded protein response will activate it, right? So it happens every day, but it's also very important for pathological condition as well. So if you think about diabetes, if you think about neurodegeneration, if you think about viruses, right? So for example, they all kind of link, they all link to unfolded protein response. If you think about cancer cells addicted to molecular chaperone, they usually prefer to keep UPR up, right? So to make sure that they can fold all this complex protein, in the because they always have stress, right? So this is very, very important question, and we need to understand all details. But at the same time, this is a signaling pathway. It's very important and involves a lot of components, right? And we don't understand it in all details. So again, I'm simplifying things a lot, right? So, but my lab is particularly interested in one branch of unfolded protein response called IRE1 response, right? This is the most conserved, evolutionarily conserved UPR branch in eukaryotic cells, right? And just make it very, very simplistic, right? IRE1, it's shown here very cartoonishly, right? IRE1 is a stress sensor. The problem with ER stress response that the cell needs to signal that, okay, stress happened inside the ER, it needs to propagate the signal to the nucleus, right? So it's a membrane on the way. So this is the reason why stress sensor, this is a transmembrane protein. So ER has a luminal domain, which is responsible for stress sensing. And stress in this case is accumulation of unfolded proteins, right? So then it has a transmembrane part, and then it has a cytoplasmic domain. And cytoplasmic domain is responsible for downstream signaling, and the outcome of UPR will be other expression on molecular chaperone inside the ER. Right? So it's very complex, but we have two main players here. We have IR1, which is a stress sensor, and we have a molecular chaperone called BEEP, which is shown here in blue, right? And BEEP is a HSP70 molecular chaperone. Again, it's a complex protein, it's a two-domain protein. So overall, the system very challenging. So what we have here, we have a very promiscuous substrate recognition because IR1 needs to recognize a lot of different unfolded protein inside the ER, right? So, but this recognition is still specific. We have a very trenched protein-protein interaction between IR1 and B, but between also other components. And we also have self-oligomerization of IR1, which is absolutely key for the whole response. We also have multi-domain protein which undergo conformational changes, and those conformational changes coupled with each other. So again, this is the next level of complexity. And then we also have post-translational fine-tuning, and we have a feedback regulation in the system. So we have a multiple level of how we cell control this system. So taking this into account, the next question will be, is NMI good enough to characterize this? So why it could be not very good? Because we are talking about large protein. We are talking about protein which are larger than 50 kilodalton. We have a lot of structural and oligomeric heterogeneity in the system. And we have to work with this because this heterogeneity is absolutely crucial for function, right? So we have limited long-term stability. Those proteins are not very stable, but they're also the concentration, the working concentration, especially for R1 is like less than like micromolar or sub-micromolar range, right? So, and of course, we expect a lot of low sensitivity, a lot of spectral overlap, right? So in this case. But because I'm here, so you probably know the answer to this question, right? So it's actually NMR is good, right? So, but we need to take all this into account because otherwise we're in trouble, right? So otherwise it's just the system is too difficult. So we need to be careful. We need to do a lot of kind of thinking and experimental design, but also construct design as well. Okay, and the first question, I mean, I only have like 30 minutes, less than 30 minutes for this, right? So I'm not going to cover everything, but I will discuss three key questions very briefly, right? So then the first question will be, how unfolded protein and BEEP control IR1 oligomerization? So IR1 oligomerization is the key process for UPR activation. So usually then we're talking about UPR, IR1 linked UPR, we are talking about oligomerization of the luminal domain of IR1, and then we also have oligomerization of cytoplasmic domain after that. So we do know that the unfolded protein substrate trigger this process, right? So without unfolded protein, it's IR1 is monomeric. Then we have accumulation of unfolded protein inside the ER. IR1 became oligomeric, right? So we do know this, and we also know that BEEP has a negative control, right? So it's like, you know, BEEP, if we have enough BEEP will prevent IR1 from activation, oligomerization and activation, right? If we don't have enough BEEP, because BEEP is too busy with unfolded proteins, right? Because it's a molecular chaperone, it's the key function is interacting with unfolded protein. So if we don't have enough BEEP, IR1 will oligomerize, right? So we want to understand how these two factors, accumulation of unfolded protein and ability of BEEP to interact with IR1 would control IR1 oligomerization. And we have to work with simpler system than the full-length protein in vitro, because this is a transmembrane protein. It's very difficult to characterize it in vitro. So we can just cut luminal domain and try to characterize it, try to see how it's oligomerized depending on different factors. So in this case, we also have this region here, which is a just a membrane segment, and this is about like 60 residue intrinsically disordered region, and we decide to keep this region because actually protein behave better if we have this intrinsically disordered region together with the core luminal domain, right? So this is our kind of a minimal functional unit, we call it, right? So this is a still kind of, it's not a full-length protein, but this is suitable for in vitro characterization for NMR, etc. Okay, so what we can do, we can actually characterize the system using different techniques. We can see how unfolded protein will trigger IR1 oligomerization, and it's very, we can use many different techniques, and we have used many different techniques. I just want to show the result for dynamic light scattering in this case. If we have like roughly 5 micromolar IR1, we add this peptide, which mimic unfolded protein substrate. So we don't want, at least for this experiment, we don't want to have the full length unfolded protein substrate, just to simplify again the system a little bit. So we have this model substrate. So we add, so this is our IR1 without substrate, right? Without this unfolded, without this peptide. It's a dimeric in solution at this concentration. We add this peptide and it's became oligomeric, right? So we can see the formation of large particles in solution. And the size of these particles depend on the IR1 concentration and the substrate concentration. So if we add more and more, if we increase IR1 concentration, we add more and more substrate, right? So what we will see that our protein became insoluble. So the oligomers became so big that they became insoluble, right? So if your protein crushed from solution, your first thought probably, okay, maybe I have some destabilization, maybe it aggregates, maybe something bad happened, right? And this is one of the reasons why we ask Joseph to do solid state NMR for us for this pellet, right? So because we want to make sure that we don't have aggregate, we don't have misfolded, unfolded protein in solution. And this is actually solid state spectra of this aggregate, right? So this is an IR1 in the presence of with a peptide, right? So, and you can see here, you see two types of spectra. So you have a CP spectra, right? Which very good for showing rigid region. And you can see very good dispersion, the resolution pretty poor, right? We don't have a specific labeling on labeling in this case, right? So we see all peaks. This is a NH spectrum. You see, but you can see very good protein resolution in this case, right? It's probably suggests, and at the same time, that if we have a NEP spectra, we only see a few peaks, and the NEP spectra particularly sensitive to flexible region. So what we conclude that in this precipitate, we actually have folded protein, right? So we have folded protein with some unfolded bits, but it's mostly folded, right? Then we did cryo-EM. No. Then we did cryo-EM, and the photo is not very good, so sorry for this. But we can see, and it's very consistent, we can see very long oligomers, right? So very long fibril-like particles, right? And if you measure the dimension of these particles, it's actually agree with a model if you put these dimers together, like in a line, right? So the dimension of these fibrils will be consistent with this dimeric model, right? So what we believe that, IR1 form this long fibrils, right? So it's oligomerized upon the peptide binding, and this is what we have, right? Then the interesting things start, then we add beep. So this is all introduction. You will need it to understand some NMR bits later, right? So if we add beep, right? So what we see that our oligomers became soluble again. And it doesn't really matter the order how you add it. So you can form oligomers, you can precipitate your protein from solution. Then after that, you can add 20 micromolar beep. You add ATP because beep require ATP as a chaperone, and it became soluble again, right? So if you decrease beep concentration, and instead of 20, you have 4 micromolar beep, 1:5 ratio, it still worked, right? So then if you decrease your beep concentration 1,000 times, it's still properly soluble, right? So it still works, right? So it's very nice. It's meaning that we don't have one-to-one complex for sure, right? So which prevent R1 from oligomerization, right? So BIP acts more like a chaperone. It's very similar than BIP interact with unfolded protein, for example, right? So you don't need a lot of BIP because it's a very, very dynamic straight-net interaction. So BIP kind of a bite unfolded protein, prevent it from misfolding and then dissociate again. So it's a very dynamic motion. So looks like something similar happens, right? And an agreement with this. So if BIP is inactive, we don't have this effect anymore, right? So R1 insoluble. So what it means inactive in this case, if we don't have ATP, BIP require ATP for function. This is an ATP-dependent chaperone. We don't have disaggregation. So if you have mutation like this, which is prevent BIP from function, right? So from being active, we don't have this, again, disaggregation anymore, right? So BIP does something very interesting with the system. It looks like it acts as a chaperone with a properly folded protein and prevents the R1 from oligomerization. Okay, so the next question we would like to address, how it's happened, how BIP can interact with a folded protein. Because BIP, again, this is a molecular chaperone, it requires exposed hydrophobic region to interact with, right? So we want to know what has happened, right? And this is actually summarized here, right? So BIP selectively recognizes R1 oligomeric species somehow. It interacts transiently, and it's requiring ATP for this, right? What does it mean? It means that it's recognized somehow solvent exposed hydrophobic region, which is only exposed in oligomeric form of R1. Okay, so if you remember, I showed you the spectra before. It's a solid state spectra of R1 oligomers, right? So nice dispersion, folded protein. So to maybe younger part of our auditorium, right? So what would you expect? How many peaks would you expect to see in solution NMR spectra of monomeric or dimeric R1 luminal domain? So this is a dimer. Each monomer is 44 kilodalton. How many peaks would you expect? Any guesses? The youngest member who wants to answer? Anyone? No, there is no wrong answer. So 44 kilodalton protein, right? So in solution, solid state looks nice and dispersed, right? So monomeric or dimeric form. How many peaks? Huh? 450. Okay. So this is what we thought, right? This is what we thought, right? So and this is what we got. Right, so this is the kind of this is one spectra. It's this guy, right? So without this, the blue spectra was like without this flexible 60 intrinsically disorder part, right? And and the black, it's the full length, right? So this is what we have. So we thought, something strange, right? So it's like, of course, when we animal spectroscopies, we do know that condition really matter, right? The sample really matter. So what we done next, we play with condition, we try different buffers, different pH, different temperature, almost everything, right? So yeah, we can improve it a little bit, not too much, right? So we play with construct related to the question we have, before, right? So maybe it's dimerization, maybe it's oligomerization, right? So we mutate protein in many possible places, right? So we stop dimerization completely. We do now, we don't have dimer anymore, right? So we have kind of now more or less homogeneous population, right? So we still have significantly less peak than expect, right? So what we can see, the folded region are not visible, right? So for R1, right? So this is another kind of example. Then maybe we have microsecond, millisecond exchange heterogeneity for the system, right? So we can't, at least so far, right? So we can't see folded bit of the protein. But the good news is that we can actually see this intrinsic disorder part, C-terminus, and we can see this oligomerization interface. And why it's important for us? Because actually, for this protein, we have X-ray structure, high resolution X-ray structure, everything is fine. But X-ray structure only show this gray region, right? In X-ray structure, this oligomerization interface completely unresolved, right? So we can't see it. And this bit, they completely chop off, right? So they don't care about this bit. The good news that, I mean, in NMR, we can actually see this oligomerization interface, and we can understand hopefully better, right? So what happened. So then we use sequence conservation analysis, right? So because very often it can also give us some idea about functionally important region in a protein. And because we are talking about disorder region, so a conservation analysis could be very, very useful, because in disorder region, we don't have evolutionary pressure on folding. So whatever conservation we see, it usually means that we have a functionally important residues, right? So it could be that they're important for post-rotational modification, for interaction, etc. So usually intrinsically disorder parts or disorder parts in the protein, the conservation level is quite low, right? So we have a good idea why, right? And this is for C-terminus intrinsic disorder region in R1. This is what we observe, right? So we have a lot of low conserved residues. We have some high conservation and some beats, which probably suggest those residues are important, right? So we probably expect something like that for oligomerization interface as well, because it's also disorder, right? And this is what we have for oligomerization interface. So actually oligomerization interface looks more like a properly folded protein, because we have a lot of conservation here. You know, we have some relatively low conserved region here and there, but overall it's very, very conserved, right? Then, of course, we also try AlphaFold, right? So because everyone doing AlphaFold these days, right? So what we did, because we have like more than 100 sequences for different organisms, right? So we run AlphaFold for all of them to see what actually happened, how AlphaFold would predict the structure. And actually it's predict these parts of the protein very nicely, very consistently, almost no differences between the organism. But if you look at the oligomerization interface, it's very different, right? And we have example, then it's completely disorder, right? We have example, then we have quite a lot of structure, and we have something in between, like it's actually in human, right? So we have partially disorder, partially folded bits, right? I should highlight here that the accuracy of AlphaFold prediction very low, suggesting that we have some intrinsic variability in this region, right? So which make it very difficult to predict, right? So some unique features there. So because our NMR for this region looks quite disorder, we decided to add a little bit of RIA, right? So because what you expect if you have completely disorder region, right? So you add the RIA, nothing should happen, right? Because, you know, it's already unfolded. So we use 0 to 0.5 RIA, and the reason for this is because we also did CD, and we do know that protein remain mostly folded up to 0.5 RIA, right? So we kind of expect that the other part of the protein mostly folded, we mostly perturb this oligomerization interface, right? And what you can see here, we just change RIA concentration from 0 to 2.5, and what you can see that for some peaks, nothing actually happened. Right? For the majority of C-terminal intrinsically disorder region, nothing happened. This is exactly what we expect. But for this oligomerization interface, for many peaks, we have this very nice peak walking pattern, right? Suggesting that we have some conformational transition, right? So then we add in a RIA. I mean, we have all this shift to the middle of the spectra, right? So in this direction or in this direction, suggesting that we have, of course, unfolding, right? Exactly what we expect. So then we actually did our solubility assay in the presence of urea. Again, we add peptide, right? So we precipitate our protein, we precipitate our R1, and then we repeat this experiment at different urea concentration. And what we see, we add urea, we don't have precipitation anymore. So what we can conclude from this, that we have highly dynamic oligomerization interface, but we still have some residual structure there, right? And this region it's neither folded or completely unfolded, right? So we have some interesting conformational variability in this region, right? And we also know that playing with this conformational variability, playing with folding and unfolding transition, we somehow interfere with oligomerization, right? Just doing this very simple two-dimensional experiment, we can actually learn quite a lot about the system, right? Why this information is important for us? Because now we want to understand how BIP interacts with folded protein, right? We do know that BIP can only interact with exposed hydrophobic region, and this interface is ideal BIP binding site, right? Because it's exposed, it's partially unfolded, right? It's a very dynamic. So there are quite a lot of hydrophobic region in this residues in this region. So this is an excellent binding site for BIP. So then what we did next, we used this algorithm called BIP-PRED. It's actually like a computational algorithm which allows to predict BIP binding site in any protein of interest. So we ran this algorithm for all residues on this oligomerization interface, right? And we found 6 possible candidates, right? We found six fragments, 6 peptides, which could potentially bind to BIP. So then we did characterization, then we did experimental confirmation for this peptide, and we used methyl NMR, and I will show you example a little bit later. Now you should just trust me, right? So we use methyl NMR, and we confirm that 2 residues or 2 regions, like here and there, they bind to BIP as a substrate, right? So now we know that, this is very schematically shown here because, it's actually this blue guy and it will be this part of the protein, right? So you can see if you have transition between folded and unfolded conformation here, so this region will be exposed to the solvent differently, and this allows BIP to bind to this region only at specific conformation. Okay, so then the next question is, I still have 10 minutes, right?
The other speakers were very well disciplined, so we are good to go.
Okay, I will try to be as quick as possible, right? So then what we did, because now we know that BIP interact with IR1 oligomeric interface, right? So now we want to ask the next question, whether the BIP fine-tuning, if we fine-tune BIP chaperone function, whether we can fine-tune somehow IR1 oligomerization as well. So what we did next, we do know, like we characterized this a while ago, that BIP function rely on conformational changes. Like BIP is a 2-domain protein. It has nucleotide binding domain which binds and hydrolyzes ATP, and it has a substrate binding domain which interacts with unfolded protein substrate, or in our case, with oligomerization interface of IR1, right? So how it's bind and release substrate depend on the nucleotide present in the nucleotide binding state, right? So if we have this conformation, it's the substrate binding is very weak. If we have this conformation, substrate binding is very strong, and we have this kind of a binding motion, right? So BIP interact with the substrate, release it, interact, release it, interact, release it, and the kinetics of the cycle depend on ATP binding and ATP hydrolysis, right? So now what we try to do, we try to see if we fine-tune this cycle, what happens with IR1 oligomerization, their oligomerization. So of course, this is a big protein. This is a 70 kilodalton protein. We can't use a MIT NMR for such system. So we use methyl NMR, so we specifically label methyl group only. In this case, only isoleucine. Again, if you're working with a big system, I would strongly recommend you personally. I mean, some people would disagree with me. Use isoleucine because they have a perfect dispersion and they have a perfect relaxation property at the same time, right? So if you look at the valine leucine, you have more overlapping, they kind of clump together. If you look at the alanine, it's also good, but then you have for the big system, they not as sharp as isoleucine, right? So for isoleucine, 70 kilodalton protein, right? So you can do as slow as 1 micromolar sample, like, you know, the spectra looks super. And you usually have quite a lot of isoleucine. They will show, for example, for BIP, they will show in black, right? So we kind of, you know, see individual peaks here. I show full-length protein in black, and I show nucleotide binding domain in blue. They nicely overlap in apostate, but in apostate only. So it's kind of, you know, it's very nice NMR spectra for very complex system. The good thing about this is that we can actually do very detailed characterization for the BIP chaperone cycle, right? And I don't have time to explain this in detail, but what's important here that using metal NMR for system like this, we can actually map each individual step. 5 minutes, perfect, I almost done, right? So we can map each individual step. We can get fingerprints for each individual step of very complex cycle or very complex conformational changes or very complex signaling system, right? So we can do each step very often for the system like this, for example, for BIP, only this and this, it's a major population step. So then we need to be clever enough, and then we need to do a construct design to make sure that we can actually trap different steps and characterize them, right? And then we can also apply CPMG, we can apply more sophisticated techniques to extract the same information from the full-length protein, from the wild type construct, right? The good thing about this is that we can actually see all these peaks at the same time, right? And here I only show you 4 peaks, but, you know, just for simplicity, but you can see each of them has unique peak position for each individual conformation. And then we can do like a mapping on the structure, on your AlphaFold structure, on the real experimental structure. And then you can also map the landscape as well, right? This is what we did. And I will skip it. So again, we use conservation analysis, right? And we find 2 post-translational modification sites on substrate binding domain of BIP, which only conserve for ER HSP70, but not for cytosolic 1, right? So we have a lot of different HSP70, right, in cytosol, in mitochondria, in ER. So if you look at the site which only conserve in ER, and we found these 2 sites, and I just show here, for example, for this site, so we have T in this position, we have G for cytoplasmic IR1, or for cytoplasmic HSP70. So we mutate those residues, and what is interesting, that those mutations, just to mimic phosphorylation, right? So we introduce D, so just to mimic phosphorylation. So what we can see, that both mutants make IR1 more soluble. This is again the solubility assay. You add BIP and IR1, your protein, your IR1, you add BIP and ATP, your IR1 became soluble. You don't have ATP, it's not soluble. So if you add either this or this mutant, it helps a little bit, but twice less than wild type, right? Somehow playing around these sites, right? So would interfere with the BIP ability to de-oligomerize IR1, right? So this site around the peptide region, right? The first explanation would be, okay, they do something with the peptide binding, right? So because they're so close to the peptide binding site, this is where IR1 oligomeric interface will bind, right? So it's something about this interaction. Actually, we can use, again, methyl NMR to monitor this, because we can measure the peak intensity for different conformation, for the main dock conformation and dock conformation. We can see whether this specific protein or this specific construct bind peptide or not. And a pH bind peptide pretty nicely. So because if you have a lot of domain dock conformation, right, in the absence of peptide, the conformation will shift, then we add peptide. So for blue one, we have something like that. For green mutant, we have something like that. So they can still bind peptide, right? But if you can see some increase here, each of these mutants increase the population of this conformation slightly but significantly. right? So it's not just simple interference with a peptide with a substrate binding. It's actually this mutant change conformation, and we can use NMR to see this. And what is more interesting that this change in conformation not only affects the ability to bind to the peptide and deoligomerize RE1, but it's also changed the rate of ATP hydrolysis. So we see the allosteric effect, somehow mutation in this part of the protein affect the rate of ATP hydrolysis in the nucleotide binding domain. So we think that this post-translational modification change equilibrium between this domain and dock and undock conformation. It's changed the protein landscape, right? But it's also affect the hydrolysis, and by doing this, it's fine-tuned beep function. So this is my last slide, right? So what we learned so far is that unfolded protein induce remodeling of dynamic oligomerization interface, promoting RE1 oligomerization, at the same time exposing beep binding hotspots. Then beep act as ATP-dependent chaperone that promote RE1 deoligomerization by interacting with this hotspot. And this is very important because it's return RE1 to the normal condition, then we have a lot of beeps around, right? Beep around. And then beep RE1 desactivation can be fine-tuned through beep, right, allosterically. And I think this, and this we learn for the complex system like this, combining different techniques together, right? And this is what we have so far. So this is all, my collaborator, not only for this project, these people did done the work, right? So this is, this is they kind of their result. And just 1 second. We have a position for the different system. We have a position in the lab. So if you're interested, right? So it's between Anton Schiner-Rasford and my lab, right? So if you're interested, please let me know or email me or Anton and Schiner, and we can tell you more about this. Okay, good.
Thank you, Anastasia.